eukaryote.
Right.
It's barely a cell in the way we think of one.
Right. A bacterium is, well, it's essentially a messy bag of DNA. It has no nucleus. Its walls are made of these weird sugars we don't have. You can poison it pretty easily because it looks nothing like a human cell.
It stands out like a sore thumb.
Exactly.
Yeah.
But these things, fungi, parasites.
They have a nucleus.
They have a nucleus. They have mitochondria. They have the same cellular machinery as the person they are, well, killing.
Which makes them the ultimate guerrilla fighters.
They are wearing our uniform. And that biological similarity is the root of the entire medical crisis we're about to unpack. Because if you shoot at them, you are, in many cases, shooting at yourself.
That is the similarly terrifying realization that serves as our hook today. We're doing a Science First deep dive into Module 6. And Science First means we aren't just listing symptoms. We're not just saying this bug gives you a tummy ache. We are looking at the machinery.
Because if you don't understand the machinery, the cellular structures, the metabolic pathways, all of it, you just cannot understand why these infections are so notoriously difficult to cure.
So let's start there with that clinical implication you mentioned.
Yeah.
Because they're eukaryotes, just like us, they have ADS ribosomes, they have a cytoskeleton.
Correct. All the same internal hardware, more or less.
So if I develop a drug that's designed to stop their protein production,
You are very likely going to stop your own protein production. This is exactly why antifungals and antiparasitics are often incredibly toxic.
collateral damage.
It's the definition of collateral damage. With antibiotics, you can be a sniper. With antifungals, you're often using a shotgun in a crowded room. You're going to hit bystanders.
And this similarity, it also dictates how our own immune system reacts. Right. I mean, it can't be a straightforward fight.
No, it's not. It's a total mess. Because they are so similar and often so physically large and complex, the immune system has to work much, much harder and differently to recognize and eliminate them.
Which can lead to what? Chronic infections.
Chronic infections where the bug just stays put for years, or, and this is the really insidious part, cases where the host's own immune system causes the majority of the damage while trying and failing to fight off the invader.
So the mission today is to unpack this complexity from the ground up. We are going to move systematically. We'll look at classification, then structure, metabolism, the life cycles, which are just wild, and then finally immunity and therapy.
It's a full journey from the outside in. Let's do it.
All right, let's start with section 1, classification, sorting the invaders. We should probably start with the fungi.
Okay, fungi. They are their own kingdom, of course.
Yeah.
But clinically, for the purpose of human disease, we really only care about two main architectural types.
Okay.
You have the yeasts and you have the molds.
Yeasts and molds. Simple enough, but let's get specific. What's the difference, really, under the hood?
Think unicellular versus multicellular. That's the big dividing line. Yeasts are unicellular. They are single little cells, and they reproduce by budding.
Budding.
Yeah, if you look at a diagram of a yeast, you'll literally see a little daughter cell just popping off the side of the parent.
Like it's blowing a bubble of itself.
That's a great way to put it, exactly. Molds, on the other hand, are multicellular. They grow these long, branching filaments called hyphae.
Ah, so that's what makes them look fuzzy.
That's the fuzz. They look fuzzy or woolly on a petri dish because of this massive interconnected network of hyphae.
Now, here's where it gets really interesting for me. You have these two buckets, yeast and mold. But then you have this third category that sort of refuses to stay in a box, the dimorphic fungi.
Ah, the shapeshifter.
Or shapeshifters. I want to stop on this dimorphic category for a second because it sounds cool, but biologically, switching your entire body plan, your whole structure, that's incredibly expensive energy-wise.
It is.
So why would an organism bother evolving two completely different structures?
It's strictly a survival calculation, and it's all based on territory. Think about where they start. In the environment, say, in the soil, it's cold, it's nutrient poor, and it's really competitive.
So you need to forage.
You have to forage. So the mold form those long branching hyphae. That's essentially a root system.
It's a net. It's casting a wide net to get nutrients.
Exactly. It's a foraging network. It maximizes surface area to suck up whatever nutrients it can from the dirt. It's designed for the ambient temperature, around 25 degrees Celsius.
Okay, but then I'm out hiking and I inhale that spore. It lands in my lung.
And suddenly, the environment is totally different. Right. It's 37 degrees Celsius. It's warm, it's moist, it's unbelievably rich in nutrients, sugar, iron, everything it could possibly want.
But it's swarming with my immune cells, which are trying to kill it.
They are trying to kill it immediately. So that big, spread out root system is now a liability.
It's too big, too slow.
It's too big, too slow, too easy a target. So triggered by that body heat, and this is a specific genetic switch, they retract. They repackage themselves
Into yeasts.
into yeasts, little armored mobile tanks. They trade their gathering capacity for combat mobility.
So the old mnemonic mold in the cold, yeast in the beast isn't just a cute rhyme.
No.
It's a tactical gear switch.
Yeah.
It's like a soldier taking off a giant backpack to get into a firefight.
That is the perfect way to put it. And on an exam, or more importantly, in clinical reasoning, this dimorphism is a key virulence factor. It's a sign of a serious pathogen.
Because it can adapt.
Because it represents adaptive pathogenicity.
Right.
If they couldn't switch, our immune system would wipe them out in minutes.
Okay, so that's the fungal breakdown. Yeasts, molds, and the dimorphic tanks. Now let's pivot to parasites. This feels like an even broader category because, we are jumping from single cells to literal animals.
It is a massive, massive umbrella. Parasite here basically means eukaryotic pathogen that isn't a fungus. But you're right, we generally divide them into two huge structural categories, protozoa and helminths.
The tiny and the long.
Precisely. Protozoa are unicellular eukaryotes, so single-celled again. They are microscopic. Think of things like malaria, which is the Plasmodium parasite, or toxoplasma. You absolutely need a microscope to see them.
And helminths.
Helminths are worms. They are multicellular animals. They have organs. They have tissues. They are macroscopic. You can see them with the naked eye.
The diagram of the pinworm in our source material really just stopped me in my tracks.
It's jarring, isn't it?
It really is. We spend so much time in biology looking at little dots under a microscope, and then suddenly you are looking at an animal, a literal animal that lives inside a human. It has a mouth.
It has a mouth. It has a muscular esophagus. The notes even point out this bulb structure in the esophagus.
What's that for?
It's a muscular pump. Think about it. It needs to actively pump food into its gut against the pressure of its own body.
And that leads into this long, winding intestine. But look at the middle of the body. It's just packed with a reproductive system.
Oh.
You can see the opening of the vagina. The uterus is just stuffed with eggs. It's an egg factory.
And the tail is pointed, hence the name pinworm. You can distinguish the anus from the vesicle at the very tip.
But here's the so what that I want to get to. Because it has muscle, and because it has a nervous system to control that muscle, that means it has a vulnerability that a bacterium just doesn't have.
You've just hit on the absolute cornerstone of anti-helminthic therapy.
We aren't trying to poison its metabolic pathways, not primarily like we do with cancer or bacteria. We can actually target its ability to move.
Precisely. If you look at drugs like ivermectin or Parental, they don't dissolve the worm. They don't poison it in the traditional sense. They target the ion channels in the worm's nervous system.
So they paralyze it.
They induce a spastic paralysis.
So the worm basically has a seizure, a full body cramp.
Essentially, yes. The worm seizes up, it loses its grip on your intestinal wall, it can't swim against the current of your gut anymore, it lets go, and the body simply flushes it out.
That completely changes how you view the treatment. You aren't disinfecting the patient. You are evicting a squatter by knocking him unconscious.
That's a great analogy, and it highlights perfectly why understanding the structure, the fact that it is an animal with muscles and nerves, is what dictates the therapy.
This brings us perfectly into section 2, cellular structure and membranes. Let's call it the hardware. We've looked at the whole worm, but let's zoom way back into the cellular level for the fungi and protozoa.
Let's stick with the fungi first. We established they're eukaryotic, so they're similar to us, but they aren't identical. And there is one key, beautiful difference in their membrane that is the holy grail of antifungal therapy.
You're talking about ergosterol.
Ergosterol, exactly. Human cell membranes are stabilized by cholesterol. We all know cholesterol. It keeps our membranes fluid but stable. Fungal membranes don't use cholesterol. They use a very similar molecule called ergosterol.
It performs the same function, though, right? Membrane stability.
Same function, yes, but a slightly different 3D shape. And that slight difference is everything. It allows us to create drugs that can bind to or inhibit ergosterol without hopefully messing with our own cholesterol too much.
So when we talk about drugs like amphotericin B or fluconazole later on, they are essentially heat-seeking missiles for this one specific molecule.
That's the target. That's the bullseye. If fungi use cholesterol, we'd be in very, very big trouble. We wouldn't have any safe way to attack their boundary.
Now, looking at the yeast cell structure itself, again, referring to the source imagery, it's deceptively complex for a single cell.
It really is. A yeast cell has a thick cell wall on the outside. It's a huge distinction. Humans do not have cell walls.
Another good drug target.
An excellent drug target. Then, inside the cell membrane, you have this massive vacuole. In the diagram, it's filled with what are called vacuolar granules.
What are those for? Storage.
Storage mostly, phosphates, amino acids, that kind of thing. But it's also critical for pH regulation. It's like the cell's pantry and its trash compactor all rolled into one.
And the nucleus looks pretty standard, but there's a little note here about a bud scar.
Ah, yes.
A bud scar.
It's a physical mark left on the cell wall where a daughter cell previously detached after budding.
So it's like a belly button.
It's exactly like a belly button, but a mother yeast cell can have many of them. It tells you the history of that cell's reproduction. You can count the scars and estimate the age of the cell.
That's fascinating. It's a physical record of life right there on the cell wall. Okay, let's shift gears to the protozoa. Specifically, the group called the epicomplexans.
The epicomplexans, yes. This group includes some of the real heavy hitters, like Plasmodium, which causes malaria, and Toxoplasma.
And the source material gives us this incredible cross-section of an epicomplexan, and honestly, it looks like an alien spacecraft.
It really does. It's just packed with gear. I mean, bacteria are usually minimalists, right? A chromosome, some ribosomes, and that's about it. This thing has specialized hardware.
It has gear for invasion.
Yeah.
That's clearly its entire evolutionary purpose. At the very tip, the nose of the cell, you have something called the conoid and the intraconoid microtubules.
Right.
That sounds like a drill bit.
It acts like one. It's this spiraling set of microtubules that provides structural support and, we think, mechanical force for piercing into a host cell. It's the point of entry.
And right behind that drill, you have these sacs called rhoptries and mochronyms.
These are specialized secretory organelles. Think of them as the payload or the lockpicking kit. Okay.
When the parasite makes contact with a host cell, the micronemes fire first. They release proteins that help with adhesion, with grabbing on. Then the rhoptries inject a cocktail of proteins that literally modify the host cell's membrane.
So it's not just bumping into the cell and breaking in, it's hacking the lock.
It's hacking the lock and redesigning the door on the fly. It creates this special interface, a vacuole, that allows it to slip inside without rupturing the host cell right away. It builds its own little hiding place as it enters.
And once it's inside, it uses these other specialized organelles. There's one called the acetopalsisum, which the notes say regulates calcium and pH. But the most surprising feature, and one of my absolute favorites, is the epicoplast.
The apicoplast. The diagram labels it as a plant-like vacuole.
And that's the aha moment for me. What on earth is a plant part doing inside a human parasite?
This is one of the most beautiful examples of evolutionary history, what we call endosymbiosis. Millions and millions of years ago, the ancestor of these parasites engulfed a microscopic algae, a single-celled plant.
And instead of digesting it, for some reason, they kept it. Over eons, that algae lost its ability to do photosynthesis, but it retained some of its other essential metabolic pathways.
So these parasites, the ones that cause malaria, have a remnant of a plant living inside them.
They do, and that apicoplast is crucial for making things like fatty acids. But here's the kicker. Because it's plant-like, it has biology that is totally, completely different from humans.
Which makes it a drug target.
Bingo! Humans do not have apicoplasts. So theoretically and in practice, you can use drugs that are similar to herbicides, weed killers, effectively, to target the apicoplast and kill the parasite without harming the human host.
That is just wild.
Yeah.
We are treating malaria with weed killer logic because of an ancient evolutionary merger between two single-celled organisms.
Evolution gives us these opportunities if we just look close enough.
Before we leave structure, we have to mention the skin of these things. The apicomplexan is something called a pellicle.
Right. The pellicle is more than just a membrane. It's the plasma membrane plus an inner membrane complex. It's a double layer of armor that provides protection and maintains that bullet-like shape, which is important for invasion.
And for the helminths, the worms, we mentioned it before, but they have that thick cuticle.
The cuticle is essentially a tough, non-cellular hide. It allows the worm to survive in incredibly harsh environments like the hydrochloric acid of your stomach or the enzymatic soup of your intestine. It also acts as a really effective shield against the immune system. It's very hard for immune cells to punch through a thick cuticle.
Okay, so we've got the hardware down. We know what they look like. Now let's talk about how they live. Section 3, metabolism and growth. This is what the outline calls the pace of life.
And this is where we see a huge divergence in strategy. And the speed of growth tells us an enormous amount about the kind of disease they cause.
Let's look at the three groups again. Fungi first. What's their pace?
Fungi are generally slow growers, especially compared to bacteria. They're aerobic or facultative anaerobes, and because they are slower, they often struggle to compete with aggressive bacteria or a robust, healthy immune system.
So that's why fungal infections are so often opportunistic.
Exactly. They thrive when the host is down, when you're immunocompromised, or when the normal bacterial flora in your gut has been wiped out by antibiotics. They wait for an opening in the competition.
Okay, then what about protozoa?
Protozoa are the speed demons of the eukaryotic world. They have rapid replication cycles. They can replicate intracellularly inside your cells or extracellularly, and they're very metabolically flexible. They can adapt to different tissues and different nutrient sources very quickly.
And finally, the helminths, the worms.
The worms play the long game. They have extremely slow growth, and they have incredibly long lifespans. We're talking years to, in some cases, decades, living inside a single human body.
Decades. A worm can live inside a person for decades.
Decades, yes. And to survive that long without killing the host or being killed by the host, they have to have a minimal metabolic footprint. They try to be invisible. They don't want to cause a scene.
But there has to be a cost to this presence, right? Even if they're quiet.
Mhm.
This brings us to the neuroconnection that's mentioned in the outline.
This is a really critical insight. Fast-growing pathogens like bacteria causing sepsis tend to cause acute organ failure. It's a storm. But slow-growing pathogens, like many fungi and helminths, what they cause is chronic inflammation.
And the brain absolutely hates chronic inflammation.
The brain is incredibly sensitive to it. Even if the parasite isn't physically in the brain, the systemic low-grade inflammation from a chronic infection somewhere else in the body can affect the blood-brain barrier, it can alter neurotransmitter levels, and it can lead to the subtle, persistent neural dysfunction.
So we're not talking about a sudden stroke.
No, not at all. We're talking about mood changes, cognitive fog, chronic fatigue, things that are really hard to pin down.
It's the slow burn. The nervous system is basically simmering in inflammatory cytokines for years on end.
Exactly. And that takes a huge toll over time.
Moving on to section 4, life cycles, entry and dissemination. This is really the meat of the episode because the complexity here is just staggering.
The rule of thumb here, I think, is that the entry route predicts the tissue tropism. How it gets in tells you where it's going to live and how the immune system is going to see it.
Okay, let's run through the entry list. Fungi. How do they get in?
Mostly inhalation of spores. That's why so many systemic fungal infections start in the lungs. Or, direct contact skin infections like athlete's foot. Or, as we said, opportunistic overgrowth of your own flora like a yeast infection.
And parasites. Much more varied, I'm guessing.
Much more varied. Ingestion is a big one, swallowing cysts in contaminated water or food. Skin penetration is another. Hookworms can literally bore through the skin of your feet if you walk barefoot on contaminated soil. And then, of course, the vector-borne route. Mosquitoes, flies, tsetse flies, assassin bugs, anything that injects the parasite directly into your blood.
I want to do a deep dive into 2 specific life cycles from our source material because they're just so intricate and, frankly, so clever. First, Cryptosporidium.
Ah, yes. Cryptosporidium, a master of the ingestion route. It causes absolutely severe diarrhea, especially in immunocompromised people.
So walk us through the cycle. It starts with a glass of contaminated water.
You drink water that contains something called an oocyst. This is a tough, chlorine-resistant armored egg. It travels down to your gut. When it hits the conditions of the intestine, the change in pH, the bile salts, it undergoes what we call exsistation.
It hatches.
It hatches. 4 sporozoites break out of the oocyst. These are the infectious mobile units. They swim over and attach to the epithelial cells that line your gut.
Now, this is the part in the diagrams that really confused me. The source says it occupies a unique niche, intracellular but extracytoplasmic. How could you be inside something but also outside of it?
Okay, it's a great question. Imagine you're standing on a plush carpet, and then you pull the carpet up and wrap it over your head. You are under the carpet, but you aren't in the floorboards beneath it.
Okay, that visual really helps. So you're enveloped by the cell, but not truly inside its main compartment.
Exactly. The parasite attaches to the cell surface, and then the cell's microvilli, these little fingers on the surface, fuse and grow up and around it, encapsulating it. So it's inside the cell membrane, under the carpet, but it has not actually entered the cell's cytoplasm.
The floorboards.
That is a phenomenally sneaky hiding spot.
It is.
It's protected from everything in the gut lumen, so it's not getting washed away by diarrhea, but it's not fully exposing itself to the internal alarm systems of the cell cytoplasm.
That's the genius of it. And once it's in that safe little space, it undergoes asexual replication, what we call merogeny. It replicates to produce 4 to 8 new parasites called merozoites. These merozoites then burst out, find new intestinal cells, and the cycle repeats. This is the amplification loop. And that's what causes the massive watery diarrhea, millions of intestinal cells being infected and destroyed over and over.
Yes. But it's not just a dead end. Eventually, some of the parasites differentiate into sexual forms. Microgametes, which are like the male sperm, and macrogametes, the female egg. They fuse to form a zygote, which then develops into a newly sporulated oocyst, and that is excreted in the feces to go on and infect the next person.
Nasty, but incredibly efficient. Now, we need to take a deep breath before we look at the second life cycle, malaria, or Plasmodium.
The mosquito-borne killer. This one is a masterpiece of biology.
The diagram for Plasmodium looks less like a life cycle and more like a subway map of Tokyo during rush hour. It is just incredibly dense.
It's definitely the most complex logic puzzle we have to unpack today.
Let's try to trace the silence versus the noise, because that seems to be the rhythm here. Let's start with the mosquito.
An infected mosquito bites you, and it injects sporozoites from its salivary glands into your skin. These travel very rapidly through the bloodstream. But, and this is the absolute key, they don't infect red blood cells yet.
Where do they go?
They go straight to the liver. This is the liver stage.
And this is the silent part.
This is the silent phase. You don't feel sick at all. You have no idea you've been infected.
The patient just feels totally fine.
Perfectly fine. Inside the liver cells, the hepatocytes, they multiply like crazy. One sporozoite can become tens of thousands of new parasites, forming a structure called a merosome. This can last for a week, 2 weeks, sometimes longer.
This is the calm before the storm.
It's the ultimate calm before the storm. But then the liver cells rupture, and they release thousands upon thousands of these new parasites, now called merozoites, into the blood. And this is where the noise begins.
This is the part I found confusing. They don't just swim around freely in the blood, do they? They have to hide again immediately.
Immediately, within seconds, they dive into red blood cells.
So the merozoite is the form that breaks into the red blood cell.
Correct. And once inside, it changes its clothes again. It becomes what's called a ring stage. If you look at a blood smear under a microscope, it literally looks like a tiny diamond ring inside the red blood cell.
Wow.
Then it grows into a trophozoite, that's the feeder. It literally eats the hemoglobin inside your red blood cell.
It's eating your oxygen supply.
It's eating the protein that carries your oxygen. And it's growing until the cell is so full of new merozoites, at this point it's called a schizont, that it just bursts.
And that rupture, that's the fever. That's the sickness.
That's the crash. Every 48 or 72 hours, depending on the species, synchronized across billions of red blood cells. Boom. A flood of parasite toxins and cellular debris hits the bloodstream. The immune system flares up. You get the classic malaria symptoms: shaking chills, burning fever, drenching sweats, and then repeat.
And just like crypto, there has to be a sexual escape plan.
There is. Some of the merozoites, instead of replicating, differentiate into male and female gametocytes. These don't cause symptoms. They just drift in the blood, waiting.
Waiting for what?
Waiting for another mosquito to bite that infected human. If a mosquito picks up these gametocytes in its blood meal, the sexual cycle continues in the mosquito's gut, creating new sporozoites that migrate to its salivary glands, ready to infect the next person.
It's an incredible, unbelievable relay race. Mosquito to liver, liver to blood, blood back to mosquito.
It is. It's a biological machine that is perfectly designed to spread.
Okay, so the invaders are in. They're multiplying. Now, the defense system finally wakes up. Section 5, the immune response. The battle.
Let's look at who fights whom. For fungi, the source is pretty clear. The battle is dominated by the innate immune system, specifically macrophages and neutrophils.
The eaters. The phagocytes.
The eaters. Fungi are generally small enough, at least the yeast and the spores, to be phagocytosed. Macrophages engulf them. But to do this effectively, you need an intact cell-mediated immunity.
You need T cells.
You absolutely need your T cells to tell the macrophages to get angry and start killing efficiently.
And the source notes that antibodies are generally poor at fighting fungi. Why is that?
It's a great question. You can have antibodies against a fungus, but they don't usually stop the infection on their own. The fungus is just too tough, the cell wall is too thick. You need the cellular ground troops to really do the job.
And that's why HIV patients who have depleted T cells get such devastating fungal infections.
Exactly. They might have the antibodies, but they lack the generals, the T cells, to command the troops.
Now, parasites, specifically the helminths, these things are huge. A macrophage can't eat a worm.
No, it would be like you trying to eat a whale. It's just not possible. So the immune system has to use a completely different strategy. It shifts to the heavy artillery.
And this involves IgE, eosinophils, and mast cells.
Right. Here's the cinematic sequence. First, your body makes a special class of antibodies called IgE. These are specifically tailored for parasites and, as it happens, allergies. These IgE antibodies go and they coat the entire surface of the worm. They tag it.
They're painting the target for the bombers.
Exactly. Then the bombers arrive. These are the eosinophils, those white blood cells with the bright red granules we see in the blood cell lineup. They have receptors on their surface that lock onto the IgE.
So the eosinophil physically docks onto the worm.
It docks, and then it degranulates. It releases those toxic red granules directly onto the surface of the worm. It's a close-range chemical bombardment. It's releasing things like major basic protein and other casonic proteins. These are enzymes designed to literally burn through that thick cuticle we talked about.
It's chemical warfare at the cellular level.
It is. And at the same time, mast cells are also joining in, releasing histamine to increase blood flow to the area and increase gut motility, trying to physically flush the worm out.
The source material mentions DH2 skewing. What does that mean in this context?
So the T cell part of your immune system has two main modes. Th1 is for killing intracellular things like viruses or some bacteria. Th2 is the mode for dealing with extracellular parasites. Helminths are masters at actively pushing the immune system toward a Th2 response.
Okay.
And interestingly, this Th2 response is also about containment and tissue repair. The source note says something really insightful. Parasites shift immunity from killing to containing.
So they'd rather be walled off in scar tissue than wiped out in a massive, damaging battle.
And sometimes the body agrees with that strategy. Killing a massive worm inside your liver creates a huge toxic mess. Sometimes it's just safer for the host to build a wall around it and tolerate its presence.
That's a perfect segue to the sidebar here on Staphylococcus aureus. We have an image comparison.
Yeah, if you look at the S. aureus image, you see it forms an abscess or a biofilm. It's aggressive, it destroys tissue, it creates pus, it fights dirty, it's an all-out brawl.
Whereas the eukaryotic parasite?
It prefers a truce, it wants to go into latency, it wants persistence, it wants to be tolerated.
Which brings us directly to section 6, evasion, persistence, and latency. How do they survive the bombardment and negotiate that truce?
They have evolved some truly elegant mechanisms. Fungi, for example, use capsules. Cryptococcus, which causes meningitis, has this massive polysaccharide capsule that acts like a slimy, slippery shield. Macrophages literally slide right off it. They can't get a good grip to eat it.
In protozoa.
Antigenic variation is their big trick. Parasites like trypanosomes and plasmodium can change the proteins on their outer surface. So just as the immune system produces an antibody that learns to recognize coat A, the parasite population switches to wearing coat B.
It keeps the immune system constantly one step behind.
Constantly playing catch-up. And helminths, the worms.
What do they do?
Immune modulation. This is maybe the most sophisticated strategy of all. They release molecules that actively suppress cytokines. They induce tolerance. They basically whisper to the immune system, shh, it's okay, calm down. Don't attack, let's just chill.
There is a very specific, high-level mechanism mentioned in the source regarding CCR8 and TREGs. Let's unpack that because it feels like something out of a heist movie.
This is fascinating, cutting-edge immunology. So we all have regulatory T cells or TREGs. Their job is to shut down immune responses so we don't develop autoimmune diseases. They are the police that come in and stop the riot.
Okay, the peacekeepers.
The peacekeepers. Now, certain pathogens and also many tumors have figured out how to recruit TREGs that express a specific receptor called CCR8. The source diagram shows the pathogen releasing signals that specifically call in these CCR8 positive TREGs.
So the parasite calls the police on the immune system that's trying to attack it.
Yes. And when these specific TREGs arrive at the scene, they secrete signals that inhibit the effective T cells, the killers, and the antigen-presenting cells. They essentially create a no-fight zone, a sanctuary, right around the parasite.
That is diabolical. They are using our own internal peacekeeping force to create a shield for themselves.
It's the ultimate manipulation, and it's a primary mechanism that allows for long-term persistence.
Yeah.
The immune system is effectively turned off locally.
But there's a cost-benefit analysis here. The outline mentions a trade-off table from the source.
Right. It poses the question, why would the host ever allow this to happen?
Okay.
Clearance, which is killing the bug, is good because, well, you get rid of the infection. But the cost of that is immunopathology damage to your own tissues from the toxic fight. But persistence.
What's the benefit of letting it stay?
The benefit is that it protects you from that self-inflicted damage. The source also suggests it might protect from autoimmunity and allergies, which is the basis for the hygiene hypothesis. But the cost is, of course, a chronic infection and the resulting chronic inflammation.
And as we learned earlier, the chronic inflammation is terrible for the brain.
Exactly. There's a great quote here. Unlike viruses that go silent, parasites coexist. That coexistence is a negotiated peace, but we are still paying a tax for it every single day.
Let's talk about that tax, specifically the tax on the nervous system. Section 7, CNS involvement. This is neuroparasitology.
The source provides a Venn diagram that I think is incredibly helpful for anyone trying to make a differential diagnosis. It looks at three main symptoms: fever, seizure, and eosinophilic meningitis.
Okay, so let's visualize three overlapping circles. Let's go through the zones one by one.
In the fever-only circle, this is where you see things like amoebiasis, which can cause a brain abscess, or African trypanosomiasis, also known as sleeping sickness. It's a systemic inflammatory process.
Okay, then seizure only.
This is often caused by a space-occupying lesion. Echinococosis, which forms huge hydated cysts, is a classic example. It's a physical mass pressing on the brain.
And meningitis only, specifically with eosinophils.
Eosinophilic meningitis points towards Angiostrongylus, the rat lungworm.
Now for the important overlaps.
When you see seizure plus meningitis, you should be thinking about two big ones: neurocysticercosis, which comes from the pork tapeworm Taenia solium, and paragonomyosis, the lung fluke.
And right in the center, the trifecta fever plus seizure plus meningitis. That's neuroschistosomiosis.
It is. It's a really powerful diagnostic tool. If a patient comes into the ER with seizures and you run a spinal tap and find eosinophils in the CSF, you know exactly which neighborhood of pathogens to look in.
Let's briefly mention the mechanisms for the key pathogens listed here. Toxoplasma gondii.
Toxo is famous. It forms these dormant cysts in the brain. It can cause encephalitis, especially if you become immunocompromised. But what's really fascinating is how it seems to cause behavioral changes.
Right, the rat and cat story.
In rats, it makes them unafraid of cats, which helps the parasite complete its life cycle. In humans, well, the jury's still out on the full extent, but it's been linked to things like increased risk-taking and schizophrenia.
Okay, what about Cryptococcus, the fungus?
Crypto uses that massive capsule we talked about to slide into the cerebrospinal fluid, the CSF. It causes a chronic, slow-burning meningitis. It's what we call indolent. It moves slowly, but it's incredibly destructive because it builds up intracranial pressure over weeks and months.
And Plasmodium, malaria. How does that affect the brain?
Cerebral malaria. This is primarily a vascular problem. The red blood cells that are infected with Plasmodium become sticky. They adhere to the walls of the capillaries in the brain, clogging them up.
So it's like a million tiny strokes.
That's a great way to think of it. It causes hypoxia, a lack of oxygen, and massive swelling.
And finally, Taenia solium, the pork tapeworm.
This is the single leading cause of adult-onset seizures worldwide. The larva forms a cyst, called a cysticercus, in the brain tissue. And interestingly, while the cyst is alive, it's often quiet because it's doing that immune modulation we talked about.
But when it dies,
When the cyst dies, it loses its immune cloaking ability. The immune system suddenly sees it, freaks out, and the inflammation just explodes. And that irritation is what triggers the seizure.
that brings us to the core neuro rule from the source.
Yes, and it's so important. Neural damage is usually immune-mediated or metabolic, not from direct destruction by the bug. It's not the worm eating your brain tissue. It's your own immune system throwing grenades at the worm that ends up damaging the surrounding neurons.
Or is the granuloma, the scar tissue wall the body builds that ends up pressing on delicate structures?
Exactly. The response is often worse than the invader itself.
Which leads us to the final and perhaps most difficult section of all, Section 8, Therapy and Limitations.
The weapons locker.
Let's start with antifungals. The source groups them nicely by their mechanism of action.
Okay, first up you have the polyenes, like amphotericin B and nystatin. These are the big guns, the heavy hitters. They physically punch holes in the fungal membrane by binding directly to that ergosterol molecule.
But they're notoriously toxic. Amphotericin is nicknamed amphoterrible for a reason.
For a very good reason. Horrible kidney toxicity, fevers, chills. Because, again, ergosterol is similar enough to our cholesterol that there's some cross-reactivity. Then you have the azoles.
Like fluconazole, itraconazole.
Right. These are much safer. They don't punch holes. They inhibit the synthesis of ergosterol. They stop the factory from making it. They are often fungistatic, meaning they stop growth, rather than killing the fungus outright in some cases.
And then the echinocandins.
Like caspofungin. These are newer and target the cell wall, specifically glucan synthesis. These are generally very well tolerated because, as we said, humans don't have cell walls. It's a perfect differential target.
And last, alemanese.
Terbinafine, often used for skin and nail infections.
Okay, now for the antiparasitics.
A much broader and more complicated group. They're usually divided into protozole and helminthic.
So protozole would be things like anti-malarials or anti-amoebics like metronidazole.
Correct. And helminthic drugs are then subdivided by the type of worm. Anti-nematodal for roundworms, anti-cystital for tapeworms, and anti-trematodal for flukes.
But here we arrive at the therapeutic dilemma. We alluded to it with the brain cyst, but let's make it explicit.
The source calls it the neurowarning, and it's this. Rapid parasite death releases massive amounts of antigens. So imagine you have a patient whose brain is full of those tapeworm cysts, and you give a very effective drug that kills them all instantly.
They all lose their immune cloaking at the same time.
All at once. The immune system suddenly sees hundreds of foreign bodies it was previously ignoring,
And it attacks.
and it attacks with everything it has. You get a massive cytokine surge. And in the closed space of the skull, this causes acute edema swelling and can trigger severe, life-threatening seizures.
You can actually kill the patient by curing the infection too quickly.
You can. It's a well-known complication.
So what do you do? How do you manage that?
You have to be very careful. You often have to give high-dose steroids before and during the antiparasitic treatment to intentionally dampen the patient's own immune response. You have to protect the brain from the cure.
That is such a delicate, terrifying balancing act. It really underscores why this science-first approach matters so much. If you didn't know the mechanism, you'd just prescribe the pill and create an absolute disaster.
You would. It's all about understanding the interaction between the bug, the drug, and the host.
So we've covered a huge amount of ground, from the molecular machinery to the clinical crisis. Let's try to wrap this all up with the outro, the final synthesis.
The source has this great little comparison I like, the evolutionary rock, paper, scissors.
Okay, lay it on us. What's the strategy for each type of pathogen?
Okay, so bacteria win by speed. They just replicate so fast they outrun our immune system's initial response.
Makes sense.
Viruses win by information. They mutate, they integrate, they hack our own cellular code.
Got it.
Fungi win by similarity. They hide in plain sight because biochemically they look so much like us.
And finally, the topic of the day.
Parasites win by patience. They establish tolerance, they persist for years, and they play the long, long game.
And the main takeaway, the final word on how this all affects our nervous system.
The nervous system suffers most, not from the invasion itself, but from the chronic inflammation these organisms cause. It's the constant low-grade battle that wears our most sensitive organ down.
I want to leave everyone with a final provocative thought, something that was hinted at in that persistence section. We talked about tolerance. As we wrap this up, I keep coming back to that idea of the evolutionary rock, paper, scissors. We are so used to thinking of humans as the apex predator, as the top of the food chain. But in the context of this deep dive, we look less like the masters of the planet and more like a really nice habitat.
To a parasite, you aren't a person. You're an ecosystem. You're warm, you're wet, you're predictable, and you're full of sugar. You're prime real estate.
And the better our immune system gets at killing them, the quieter and more manipulative they have to become to survive.
That's the haunting part, isn't it? The most evolutionarily successful parasites aren't the ones that kill you quickly like Ebola. They're the ones that are so good at immune modulation that they convince your body to let them stay.
Which leads to that really uncomfortable question we touched on with the hygiene hypothesis, or the old friends idea. If we succeed in our quest to scrub our world completely clean, if we remove every worm, every benign fungus, every passenger we co-evolved with, Are we actually breaking the calibration of our own immune system?
We might be making ourselves immunologically lonelier than our biology was ever designed to be.
A thought to chew on next time you sanitize your hands.
Thanks for listening to The Deep Dive.
See you in the next one.